Hi guys, welcome back to my channel. I'm so glad to see you all here. So in this video, we are going to talk about downloading data from GDC Portal using an R package called TCGAbiolinks. And if you haven't heard of any of these terms, we are going to talk about each of these terms in further detail and talk about what is the use of this portal, what data we can access from this portal, and how we can download this data into R, and what are we going to do with this data in R in this video. Genomic Data Commons is National Cancer Institute's data sharing platform, and this platform is one of the world's largest cancer genomics data collections, which includes more than 11,000 patients representing 33 cancers and over 500,000 of total files. This platform contains data from huge cancer genomic initiatives and projects like the Cancer Genome Atlas, that is a TCGA, and data from Target. And the idea behind creating this platform was to assist researchers in exploratory analysis by identifying changes in cancer cells that may play an important role in cancer development. And there are various kinds of data that researchers can download from this portal, like mutation data, copy number variance, expression quantification, post-transcriptional modification, structural variance, DNA methylation data, as well as protein expression data. And this portal contains not only the processed data but as well as the unprocessed molecular data types. So this portal serves as a hub of major data types across major cancers, and it becomes extremely important for anyone who wants to leverage this portal. It becomes important for them to know how to download this data onto their system and then can move ahead with processing the data. So before I go ahead, I want to quickly walk you through the GDC portal and kind of give you a brief idea on what the portal looks like and what kind of data is available and how you can access and navigate your way through this portal. So let's go to our browser, and when you search for GDC portal, the first link should take you to the GDC portal. So this is how the portal looks like, and there are various ways to search for data on this portal. You can look for the major primary site of your interest, or you can click on the organ of your interest, or you can use the text search here by searching with using your gene of interest or the primary site or the project name, or you can utilize the tabs available here. So just quickly walking through these tabs. So the first is the projects tab, which allows you to explore the projects that are present in the GDC portal. So here you can utilize these filters here on the left side of the page, and when you click on these filters, the number of cases or the projects here will be updated as and when you click on these projects. So quickly showing you that, let's say I'm interested in projects associated with breast. Basically I'm interested in breast cancers, and as soon as I click on the primary site breast, you'll be able to see that there are certain updates that happen to the page on the right hand side here. You can see the projects that are associated that has primary sites breast, as well as various project names here that are present, along with the number of cases and the available data for each of these projects, as well as the genes that are frequently mutated or affected in these projects. You can also apply additional filters like the data category of your interest or experimental strategy to further narrow down your search. Going to the exploration page, here you will be able to specifically look at certain more details. So let us click on breast, and this time I'm interested in the data from the TCGA program. And here you will be able to see the projects associated with these filters, as well as the number of files and the number of data that are available. In addition, you will be able to see some more tabs in the right hand side of the page. You will be able to see the number of cases that corresponds to the filters, as well as the genes that are affected in these cases. You'll also be able to find the mutations that are found in these cases, and you will be able to visualize these mutations as oncogrid on their portal, where you will be able to see what kinds of mutations are present in these cases from these projects. It will also allow you to perform some analysis like set operations on their portal. as well as repository tab, which will allow you to download the data. Now this is the area of our interest where we would essentially want to download information or files from certain projects of our interest. So let's say I'm interested in transcriptomic profiling, and I'm interested in gene expression quantification, and I'm interested in RNA-seq. And again, I can move on to the cases, and here I can apply filters that I am interested in. So I'm interested in breast cancer from the TCGA program. So essentially this is building a sort of a query. If you look at the top here, it's sort of building a query of all my filters and showing me the files that corresponds to all these filters. Now if I want to download, let's say, a couple of these files, so since I'm interested in the counts file, these are all the counts files, so I'm just going to click on the cart button here. And when you click on the cart button, it basically adds these files into the cart. And once you add them to the cart, you will be able to download the cart directly. Now this makes sense when you have only few files to download. But let's say if you want like all the files, let's say if you want to download all the 1000 or 1500 files, then downloading from their website becomes a little bit of a tedious and a bottleneck process. So there are other ways to download files. You can also download their data from the GDC data transfer tool. And if you click on the GDC app, you will be able to find the documentation, the data transfer tool, and information on various things that you can do on this portal, including the ways that you can download data from this portal. But one of the ways that I find more convenient is by using an R package called TCGAbiolinks, which will directly download these files into R, and I can start processing my data from R. So that is the method that I chose. But if you prefer downloading from the GDC data transfer tool, or by using their graphical user interface by adding to the cart and downloading, you may do so. So this was a quick and a short walkthrough and demonstration of this portal. Again, this portal has a lot more utilities and data available. Most of the data present on this portal is open source, so it will be extremely useful if you figure out how to download data and leverage this data for your own analysis, as you have access to a huge amount of open source data using this portal. So talking a little bit more about the type of data that is present on the GDC portal, and these are the terms that you will frequently encounter when you're trying to download data from GDC portal. So data can be of harmonized type or can be of legacy type. So harmonized data are essentially data from many cancer projects which are processed using standardized pipelines, and they're usually aligned to or lifted over to HG38. Now, if you're not sure what HG38 is, I have previously made a video talking about what is HG38 and the difference between HG38 and HG19, and I will add the link to that video in the description section below. Now, coming back to the harmonized data, the advantage of using harmonized data is that it allows us to analyze multiple cancer types or the same cancer types across the multiple projects. Now coming to legacy data are the data that are originally generated by the original projects, which are essentially generated by different methods, and they are mostly aligned to HG19. So it's important to know the difference between the legacy data and the harmonized data, and know which one to use when you're trying to download the data from this portal for your own analysis. In addition to harmonized and legacy data are also open and controlled on GDC portal. So the open data is the data that is not individually identifiable and requires no authorization. So you can directly add the files to the cart, just as we did some time ago when I was demonstrating the portal, where you can add the files to the cart and download the cart. But control data contains individually identifiable data for which you will require authorization to download that data. So talking about the TCGAbiolinks bioconductor package, it is a package that not only allows us to perform data retrieval, but it also allows us to build a query and allow us to integrate different data types. In addition to that, it can also allow us to perform analysis like differential expression, network analysis, or survival analysis on the retrieved data. And we can also visualize data using this package. Again, it's a bioconductor package, and in the previous video I have spoken about how to install bioconductor packages. So if you're not sure how to do that, I will add the link to the previous video in the description section below. So the TCGAbiolinks package has three main functions that I will demonstrate today. So talking about each of these functions, the first is GDCquery. Now this function allows us to build a query, and it results an object which contains a summary table with the resulting files that match our filters or match our query. So just as we applied the filters and the right hand side of the page updated on the portal with the resulting cases or the files that corresponded to our filters, similarly we can do that using the GDCquery function, where we can specify the project name, the data category, the experimental strategy, or the file types. And it will return the resulting files that matches our query. So these basically would be the files of interest that we would essentially want to download and move further with our analysis. So the next function is GDC download. Once we have built the query and it has returned the object with the resulting matching files, we can use the GDC download function to download these files into our function. The downloaded data will be saved in a directory with the project name and a subfolder with the data category. This will be more clear when I demonstrate, when we move on to the demonstration and I demonstrate how to download these files. The last function is GDCprepare, and this function basically transforms the downloaded data into a usable format, into a summarized experiment object or a data frame. So the data can be extracted or can be used from these data structures and can be moved on with further downstream analysis. So these are the packages that we'll require for today's demonstration. We will use TCGAbiolinks package to download the data from GDC Portal. We'll use Summarize Experiments to access data from the Summarize Experiment object. Math tools to summarize and create visualizations for mutation data. Heatmap to visualize DNA methylation data, and tidyverse to wrangle the data. So now let us start downloading data in R using TCGAbiolinks. So the first thing that we'll do is we will load the libraries. Now that the libraries are loaded, we will first start by building a query to download the data. And as I said previously, we will be downloading three types of data today. We'll be downloading gene expression data, DNA methylation data, and mutation data. So before we do that, we will first learn how to build a query. So we built the query using one of the TCGAbiolinks function called GDCquery, and there are two parameters which are mandatory for us to provide, otherwise the function will throw an error. The first is the project and the second is the data category. So in order to get a list of projects, we can use a function called get GDC projects, which essentially gives us a list of all the projects that are present on the GDC portal. So let us just save this into a variable, the output of this function. Let's save that to a variable called GDC projects. And once I run this, you'll be able to see that you get an object with all the information about the projects that are present on the GDC portal, including the ID, the primary site, the dbGAP accession number, project ID, the disease type, and so on and so forth. Another way to get information about the projects and other information that are required to build a query, you can find that on their TCGA help documentation. So this is a TCGA help documentation, and the link of this documentation I will be putting in the description section below. So if you scroll this help documentation, you will find that there are several tables that they have provided with information on the projects, which include the disease type, the project ID, the name of the disease, the primary site, and so on and so forth. Similarly, they also provide information on other parameters like data category. So what kind of data you're interested in retrieving, what data category does it fall under, the data type, the workflow type, the platform. So there are a lot of information from these tables that you can use, and you can use these values essentially to build a query which will fetch you the relevant information. So today I'm interested in fetching breast cancer data. So the project information that we fetched using the function get GDC projects, we are just going to filter some of these columns. So the primary site for the data of my interest is going to be breast. And when I type that, you'll be able to see that there are essentially two projects which are associated with breast cancer, or essentially the cancers that originate in the breast. And today I'm interested in getting data from this project, that is TCGAbiolinks project. And there is another function in TCGA which gives you a project summary. So there is a function called get project summary, and in this you provide the project ID as a parameter. So I'm going to provide TCGAbiolinks, and when you run this function, essentially you get some information about the details in this project, like how many files are present, what are the total number of cases in this project. So there are essentially more than 1,000 cases in this project, and what kinds of data are available in this project. This is helpful when you're trying to download certain kind of data for the disease of your interest, and you would essentially want to see whether such kind of data is available for the project of your interest. So now let us build a query. So as I said, the GDC query will requires the values for these two parameters. If you don't provide values for these two parameters, it's going to throw you an error. So the project that I'm interested in is TCGA BRCA, and the data category is going to be transcriptome profiling. So I'm just going to copy it from the help document. And now let's save the output in query TCGA variable. So GDC query has finished running and it has returned an object. So recall how when we were exploring the GDC portal, and in order to get the cases or the files that are relevant to us, we applied certain filters to our search. So we checked certain boxes, and the resulting page on the right hand side updated with the cases that match the filters. So we are replicating the same process, but we are doing it programmatically. Here technically we have checked only two boxes. We have provided what project we want and from what data we want from which project, and with that falls under what category. So right now we have just applied two filters, that is the project name and the data category. So all the resulting files and the cases that correspond to this project and that falls under this data category is going to be returned. So in order to see the resulting files, we can write a function called get results and provide it with the query output object, and this can be stored into another variable called output query TCGA. So that way we can see the output of the query. So when we look at the output query TCG, you will be able to see that there are so many files, around 4,800 files being returned, and there are several cases with their accesses. There are certain which are open, there are certain which are controlled, the file names, the submitter ID, the data category, also the type. There are certain which are gene expression, there are certain which are microRNA expression, the file sizes. There's so many information here. So once we have retrieved all the files that matched our general and the most basic criteria, we can keep on adding filters to further narrow down our search and only retrieve a subset of files or the cohort that is of our interest in order for us to move ahead with the processing of the data. So now I'm going to add more filters to only retrieve the files of my interest and further narrow down my search. So let us go back to the output query TCGA object, and here I'm going to add three filters. So the first filter is going to be the experimental strategy. So I only want files that are of RNA-seq, that are basically has the RNA sequencing data. And the second filter that I'm going to add is that for the analysis workflow type, I want star counts. And the third filter that I'm going to add is that I want all the files to have open access. I don't want the controlled or the files that requires authorization, because I do not have the authorization. So I just want the files that are available without any authorization, which are open, which have open access. So, and also, just because there are so many cases, there are over 1,000 cases, I do not want to download the entire cohort. I just want to download, for this demonstration purpose, I'm just going to download like 3 files so that I can show you how this works. So just making sure that I'm just going to filter here by RNA sequencing, and I'm just going to download essentially these three files with open access. So we are going to add all of this information in the query. So I'm just going to copy the query here again, and now I'm going to add the additional parameters and the values to these parameters. So the first one is experimental strategy, and that is RNA sequencing. The second one is workflow type, and this would be star counts. The third would be access, and this would be open. And as I said that I just want to download 3 cases or 3 files, I'm just going to provide these case IDs in the barcode. So this is going to be the first one. And this is going to be the last one. So now I have added all the filters that I wanted along with the specific cases that I want to download. So now I'm going to run this query. Now that the query has finished running, and let us see what the query has returned. So again, we will use get results function, and we will provide the output object of the query. And when we run this, now we see we have information for only 3 files, because essentially we retrieved data for only 3 files. Essentially we retrieved only 3 cases that we were interested in. So this indicates that we have information on the query returned only 3 files of our interest. And now we can go ahead and download these 3 files. So in order to download these files, we use the function GDC download, and we provide the query, the object that the query returned, that is saved in query TCGA. So now when I run this, it should create a folder in the current folder where it will download the files. And once it finishes downloading, I'll show you how it has downloaded the file. The files have finished downloading, and you can see that there is a new folder that is created, that is GDC data. And when I click on that, you will see another folder that is the name of the project, that is TCGAbiolinks. And within that folder, we have another folder called Harmonized, because we have downloaded the Harmonized data. So we click on that, and within that we see the data category folder. So when we click on that, you'll again see the gene expression quantification, which is the type of data that we downloaded. And within that we see 3 subfolders, each with the file name, and within that we'll see the actual file. So it is nested within many folders with subsequent information that was present in our query, because you can have multiple queries and you can download multiple types of data from the same project or even from the same data category. So this is really convenient when you're trying to download various data types from the same project or even from the same data category. Now that we have learned to build a query, we have applied filters to the query, and we have downloaded the files. Now let us extract data from these files. Since we have downloaded the gene expression data, it would be beneficial if we could extract the counts or the TPM or the FPKM values, which could be used for further downstream analysis. So in order to extract data from these files, we use a function called GDCprepare. So GDCprepare takes the first parameter as the output query object, that is the query TCGA, and the next value is going to be a true and false value for the summarized experiment parameter. So this basically tells the function whether to return a summarized experiment object or not. I'm going to set this as true, and I'll show you in a second how to extract the counts or the TPM or the FPKM values from the object. So this will return a summarized experiment object. So we are going to save the object as TCGAbiolinks data. You'll be able to see that this object has counts data, has TPM and FPKM data. So in order to retrieve, let's say, counts data from this object, we can use a function called assay. Since this is a summarized experiment object, we can specify what kind of assay we are interested in retrieving. So I'm going to go with the unstranded counts first. And let us save this to a variable called BRCA matrix. And when you run this, you will be able to see that you have essentially a matrix of counts. And again, we have three columns here because we have extracted data for 3 cases or 3 samples. And the row names here are the gene IDs. And these are essentially the counts which can be used for further downstream analysis like differential expression analysis. So if you're interested in, let's say, FPKM data, you can just get the FPKM unstrand and you can rerun this, and now you will have the FPKM data for all the genes for these 3 cases. So now that we downloaded the gene expression data from breast cancer tumors from the GDC portal, let us go back and download some DNA methylation data. So let us build the query first. So again, we will start with GDCquery to build the query. The project this time would be TCGA glioblastoma, which is TCGAGBM. The data category would be DNA methylation. Going back to the help document from the TCGA, since we are interested in DNA methylation, we will have to specify the platform, and this is the platform that I'm interested in to get the data from. So I'm going to specify the platform. Again, the access would be open, and the type of the data would be DNA methylation beta value, which are essentially the methylation signal intensities. And let us save this to a variable called query methylation. The query has finished running. Now let us get the output of this query. And let's save this to So looking at the output data frame, here we can see that there are around 300 rows. So we have so many files that matched our filter criteria. So since I'm not going to download the entire cohort, since these are a lot of files, I'm just going to pick the top 2 files to download. So here I'm just going to add the barcode parameter, and in here I'm going to provide the name of the files, or the cases rather. And please note that these are open access files, so I will not require any authorization to access these files. So I'm going to run these, I'm going to run this query again now. Now that the query has finished running, now I'm just going to quickly run the get results command again, just to make sure that the 2 files have been fetched, or the 2 files of my interest have been fetched. So when I run this, I can see that these are the 2 files that have been fetched. And now that I wanted the files of my interest being returned from the query, I can move ahead with the download and then can move on to preparing the data, and then we can explore the data a little bit. So let us use the GDC download function to download these 2 files. Again, these files will be downloaded in the GDC data folder within my working directory. So when I click on this folder, you will see that there is a new folder being created. So now there is another project called TCGAGBM, and this is essentially the folder where I will have my DNA methylation data, because I downloaded the methylation data from the glioblastoma project, not from the BRCA one. So it created another folder. And again, it follows the same process, that it has subfolders within the folder. It has harmonized folder, within which we have DNA methylation folder, we have methylation beta values, within which we have two subfolders, and each of these folders contains the files with our data of interest. So this is where the nesting of the folders is advantageous for us, because we can download multiple data from multiple projects without them being very confusing or chaotic. And it's separated into separate folders, which makes it very easy for us to access different information from different projects. Now that we've built the query and downloaded the DNA methylation data, next we will extract these methylation values, and I will generate a simple figure that shows the top 10 probes that have different methylation values between these two samples. So let us extract the methylation values first, and we can do that by the function GDCprepare. Again, we provide the output query object, and we set the summarize experiment value to be true, and we can save this into another variable called DNA meth. So since DNA meth is a summarized experiment object, you can access the methylation values by a function called assay. And these are the methylation values. Here you can see the row values as the probe IDs. And now we'll just perform certain basic operations to get the top 10 probes that have different methylation values between these samples. So let us start by the DNA methylation summarize experiment object. Let us access their assay, that is the methylation values. Now I'm going to calculate the row variance. This is to calculate the variance across the samples. So essentially, the probes having the highest variance will have different methylation trends between the samples. And followed by that, we will order them decreasing. So the probes having the highest variance, we'll essentially want the top 10 probes having the highest variance, which essentially will indicate that they have different methylation patterns between the samples. So I'm going to set the decreasing as true, and we need the top 10 probes. And now I'm going to save this in a variable called index. So once we have these values, we can simply create a plot. So I'm going to use a function Heatmap to generate a sort of a heat map, a small heat map that shows the top 10 probes having different methylation values between the samples. So our values are saved in DNA methylation variable, and that can be accessed using the assay function. And the rows are these top 10 probe IDs. And when I run this, you should essentially see a heat map. So basically, these are the top 10 probes having different methylation patterns between these two samples. So essentially, the whole point of doing this exercise is to show you how convenient TCGAbiolinks package makes it for us to access the information, to download the information into our local system, and perform some basic operations to process and analyze our data. Now that we have built a query and downloaded the DNA methylation data, we also visualize this DNA methylation data. Let us move on to downloading and visualizing DNA mutation data. And I want to get the mutation data and visualize mutation data from breast cancer cohort. So let us start by building the query first. And again, the project here would be TCGABRCA. This is the same project where we downloaded the gene expression data from. And for the data category, I'm going to go back to the help page of TCGA, and here under the data category, now I'm going to copy the simple nucleotide variation. So this is the value that I'm going to provide for the data category because I want the mutation data. Again, the access would be open. And now I'm going to save the output of this query into a variable called query_mutation. So before I provide the barcode or the specific cases that I want to download, I'm just going to build like a general query so I can get all the files or all the cases that match these filters, and then I can further apply filters to narrow down my search. Now the query has finished running. Let us get the output of the query into a data frame. So I'm going to save the output into a variable called output underscore query underscore mutation, and we are going to use a function called get results and provide the output object. So when I open the output underscore query underscore mutation data frame, here we can see that there are so many cases that are being fetched, and it has matched the filter criteria that we have provided when we are building the query. So under the cases, again, since I do not want to download the entire cohort for this demonstration purpose, I'm just going to download specific cases. And under the cases column here, you can see that there are two case IDs that are provided with a comma. And this indicates that these are tumor normal pairs because we are fetching the mutation data. These are the tumor normal pairs. So I'm going to fetch the first four, or rather the first two pairs from this cohort. So I'm just going to copy the entire both the case IDs in the first row, and I'm going to provide it as values to barcode. So this is the first pair, and I'm going to copy the second pair. So basically I'm going to fetch the mutation data for these two pairs. So now I'm going to run it again. So now the query has finished running. Let us quickly run get results function again, just to make sure that data for these two pairs have been fetched. And when I run this, you can see that there are these are the two pairs for which the data has been fetched. And now I can use basically this object to download this data, that is a mutation data from these two pairs, and we can further use this data to visualize the mutation data from these cases. So let us use the function GDC download and provide the output object, that is query mutation. And let us go back to the files here, and going back to our folder. Here again, the data will be downloaded in the GDC data folder. And since this project is TCGA BRCA, it has been downloaded within the TCGA BRCA folder, because previously we had downloaded the gene expression data. So when I click on this, and it has nested folders, so you can go under Harmonized. And now you can see that in addition to transcriptome profiling, we see another folder that is simple nucleotide variation, where we will find the mutation data from this cohort. And within that, we have masked somatic mutation, and we have subfolders within which we'll find the mutation data from the selected cases. Now that the data has been downloaded, let us fetch the mutation data from these cases. So we use the function GDCprepare. We again provide the output object of the query. The summarize experiment will be set to true. And here I will save the information into a variable called maf. And now we can utilize certain functions from MAFTools library to read the maf, to read this output object and generate visualizations and dashboards. And I'll show you how we do that. So there is a function called read maf, which will read the output object. So basically this is the output object where it has the information, the mutation information that is fetched from these cases. And we can save this into a variable called MAFTools input. And now we can use this MAFTools input to visualize and create dashboards. There is a function called plot maf summary, where we provide the MAFTools input object. And you will quickly see what I'm trying to do here. So we'll set certain parameters here. So basically this function creates such a dashboard which basically summarizes the mutation information that is fetched from these cases. So here you will see the frequency of the types of mutation from these cases, the variant types, the single nucleotide variants, variance per sample, variance classification, also the top mutated genes in these cases. And this is very helpful when you're trying to summarize the mutation information across a cohort. Since we have just 2 samples, this is the information that is summarized from the 2 samples. But if you have the entire cohort, this is a very informative dashboard which can help you gain useful insights from your cohort in terms of specific mutations. I'm just going to add certain comments here. In addition to creating dashboards, you can also create an oncoplot. So we will use the function oncoplot. Again, we provide math-tools input object and we set certain parameters. And this function essentially creates an oncoplot. Here the columns indicate the samples, the rows are the mutated genes, and it also shows the type of the mutation that is observed in these genes along with the frequency and the tumor mutation burden. Again, the point of this exercise was to demonstrate how TCGAbiolinks package makes it so convenient for us to access and download the data from the GDC portal, as well as fetch the data from these downloaded data so that we can analyze and process and visualize the data that we have downloaded. So that brings me to the end of this video. I hope I was able to explain how to use TCGAbiolinks package to download data from the GDC portal. I will be adding the links to various resources as well as the link to my code in the description section below. So if you found this video helpful and informative, please make sure you hit the subscribe button, like the video, share it, and leave your comments under the comment section. Until next time, see you.